Okay. Okay, got it. Yep.
Greetings, everyone. It's been a while since you've seen me on YouTube, and I'm joined here by Eric, and we're just going to get acquainted. He reached out to me. So, Eric, we kind of met through email. How did you end up finding me?
Well, I reached out to I follow this literature, so I go on the the ID streams like Discovery and Institute for Creation Research, and uh, and then I saw that Rob Stadler, who wrote a couple of books I really admire, lives in my town of Minneapolis. So I decided to reach out to him and said, "Hey, wanna have lunch or something?" And then I was working on a little idea that I have in the ID space, and thought he'd have some comments on it. So then I just stumbled on him a couple of weeks ago and said, "Hey," and then the thing I sent him, he said, "Well, that's really not his shtick." So, and I said, "Well, maybe you could find somebody." And he said, "Yeah, maybe Sal Cordova." Okay, uh, you know, just like to reach out and see what's going on. I mean,
Okay,
I'm
so
an amateur participant in this debate. I'm mainly just a follower.
all right, so I've been a volunteer and also professional in this.
Mhm.
I work um, um, for John Sanford. Now, that's an interesting story because he was an atheist. He's a famous geneticist at Cornell, Ivy League tenured professor,
Right, right, right.
and he became a Christian. He still believed in evolution, and he didn't know of any other views. Then he stumbled on Michael Behe's Darwin's Black Box, and that started him down a 10-year journey where he went from being a theistic of from atheist to Christian to theistic evolutionist to ID proponent, old Earth creationist, now young life creationist, young Earth creationist.
Yeah, it's pretty much
Um,
my journey. Although I didn't start as a Christian, I started on we have a creator. That took me about 10 years to concede. And but then that opens the door, and then you just travel down the little path. And
so that was, I just wanted to say that was a really a wonderful thing that I saw in your email. And that's why I really was hoping you could share a little bit of that with the audience. So John Sanford, ever since Michael Behe started his start on this whole question of origins was Michael Denton.
Mhm.
And then Michael Behe, the biochemist, came in, but now it's just unreal. There are so many biochemists, population geneticists, top tier chemists like James Tour,
Right.
and the number is continuing to grow. I'm seeing them at both secular and Christian universities, and tons of engineers. So things have changed, and some of
Yeah.
the stuff that we may see now. There's so many different ways of approaching the question of intelligent design and creation, and it's moving forward so fast I cannot keep up. And the quality has really improved. A part of it is not just us. A lot of it has to do with uh, experimental and observational evidence that we didn't have available to us even 10, 20 years ago. There are
Right.
arguments we're making now that we definitely couldn't have made 100 years ago, definitely couldn't have made 50 years ago, maybe even 10 or 20 years ago, even 5 years ago. Some breakthroughs have happened. They're just like, you guys are really making, that is the Darwin side. I saw that you guys are really making my job easier every day.
Right.
Um, and so the quality of the arguments, yes, it's due in part because of the growing talent pool and our skill and determination. But I'd say a lot just has to do with just the data available to us that wasn't there. And it's really causing a lot of embarrassment for the pro-evolution
Yeah.
side.
It's funny that the evolutionists keep referring to like the modern synthesis that was all before we really understood the digital nature of the genome. And I think that really just changed everything. I mean, I think they did have a viable hypothesis when you thought of it, well, why isn't it just like breeding dogs? And, if we can get Chihuahuas out of Great Danes, why can't we just extrapolate that infinitely? And if you don't know, I think Behe has said, if you don't understand something, it seems like, why not? It seems possible. And it's like Origin of Life. They thought it'd be simple because no, they had the word protoplasm for it because they just figured, well, maybe it just forms like salt crystals. And but then that's what Behe's book really opened my eyes on was you look at the digital nature, not just of the DNA, but like how proteins are made and how they fit together. And then, once you understand that, you're like, holy crap, that's going to be hard to explain bit by bit. Um, I can see us getting alleles for white skin, as you go north, because it's not really digital. It's just like, you switch out more and more alleles for less less melanin. And understand makes perfect sense, but it doesn't make ATP synthase. It doesn't make the topoisomerase or something like that. That just like that doesn't happen bit by bit, you know.
That,
So, but you have to understand, by reading Behe's book and understanding, then you realize, oh crap, the protein, like I don't know what the number is, but like if you got 300 aminos, the probability that it's just like a disordered protein is going to be probably like just randomly, it's probably going to be like what 1 in 10 to the 12th just by itself, just like it's not even going to be folded, and then so your average protein, as it tries to move over to like some other function, it's got to go through this fitness cliff valley that just in the modern synthesis they just had these nice smooth little hills, and that's the way I understood it, and it was just like, yeah, you just move, it's like hill climbing in statistical models. um but you can't hill climb when in that protein space. And then not only do you need the protein, but then you need the helpers. You need like the things that like build it and put it together at the same time, and move it around the cell. And it just when you just add up all the probabilities, I'm like, because I was a secular humanist my whole life, but actually, I'm 58 now. When I got out of college, I can see my first what is it, resume or something. I put on my describing myself as a secular humanist. So I was very proud. And I read all the Dawkins stuff with great interest, and I thought it was fabulous and really made a lot of sense to me. But just it's one of those things that makes sense at 30,000 feet. Like the homology argument. I have two bones in my hand, so does a bat, and so does a whale. So obviously that means we evolved from a common ancestor. But then you learn about convergent evolution, and then you learn about all these things having to be explained by horizontal gene transfer. And then, like, wait a second, these arguments that I thought were so convincing, they all started to fall apart. And then as I followed the argument for 10 years, I realized that there was a lot of bad faith in the rebuttals by Dawkins and his crew. And they just started like, you shouldn't be using mockery. That's in normal circumstances, you would say that's ad hominem and that's not. But they'd say, no, we're encouraging it. That's a good thing. We want to mock them. Wait, that's no, that's you would that's against your earlier principles. And but it took a long time for me to really accept it. But once I did, the cool thing about it is once you know the truth, things become clearer. And what I love about this field is that like the more I learn, the clearer things become, the more things make sense. And not just in biology, but like in my theology, learning about Christianity, the more life made sense to me, the more, the way, I thought about meaning and purpose and why they're important. And everything started to make more. I have a PhD in economics, and I started out as a macroeconomist. And there's a field where the more you learn, the less you understand things because, the models really don't work. Macroeconomic models, especially, they just don't work. They were worth a shot, but the more you learned about them, the more you learned that they didn't work. And so it's just like, oh crap, that's a lot of things. But not this. That's why I find it just so fun.
well, thank you for acquainting me with your background. I could tell you you definitely had some math background. So let me fill you in a little bit. Did Rob tell you anything about me?
No, just that you were doing some work on it, and I knew you did some work with Sanford, and I've read his work and I love it, and so I don't know what you're working on or
Okay,
what your
so
background is.
uh, so I worked for Dr. Sanford for 7 years. We're still colleagues. I'm just no longer his paid employee after working with him for 7 years. We got in peer-reviewed publications, Springer Nature, which
Mhm.
is pretty very respectable, and we got on university library shelves, which was a miracle. Now you
Well, the nice
mentioned,
thing is COVID has exposed the peer review scam that they bludgeoned ID with for so long. It's not the imprimatur of truth that they presented it to be.
but
Um,
we were able to break through. So,
hmm.
which certainly we didn't pass the review of evolutionary biologists. We went to mathematicians who actually know what they're doing. Um,
Yeah,
and
if you don't have your skin in the game, you're like, yeah, this doesn't make any sense. If like you
yeah,
don't care about like the bigger picture.
so the route that I've been telling people, I said, look, the evolutionary biologists, even ones sympathetic to you, are like, well, look, I think closetly I agree with you, but I don't want to lose my position.
Yeah,
I'm an editor. And you know what? I'm not calling them cowards because I totally respect that. There have been ID proponents in tough positions. I said, look, don't out yourself right now. We need you there. And we
right.
totally get it that, you might be an editor and you're afraid to publish ID work. And sometimes we've had to tell them, hey, look, this is not the time. You get tenured, you get famous, then speak all you want.
Right. Yeah,
That
it's just
might,
it's a shame what Lehigh did. Like, didn't they post like a, a, a declaration that they're against Behe? And it's like, why would they feel obligated to do that? That's just so
right.
counter to the whole idea that you know, they're supposed to
Well,
be just looking for truth.
so the point was we got through peer review, which was a major miracle. The editor actually approached our team and said, write on anything you want. He had that much respect for Dr. Sanford and Dr. Basner. The reason that happened is they made a publication on Fisher's fundamental theorem of natural selection,
Yeah, that was
criticizing.
great. That was a great paper. Really like
Well,
it.
it set the record by like a factor of 4 for all of the downloads in the journal Mathematical Biology, and that's also a respected journal.
Yeah.
And I think the editor took note and said, "This is the most looked at paper in that entire journal. I think I ought to invite Bill Basner and John Sanford to write on anything they want." And then Bill approached me. He said, "Sal, I'm kind of busy. Can you write the paper?" I said, "Okay, I'll start." And then we got joined by Alla Hoster, who's an award-winning mathematician in Sweden. He won the nation's highest award in mathematics. He's also a population geneticist.
Oh, well,
So you have a world famous Cornell genetic engineer in John Sanford, who invented the gene gun, and his invention's the Smithsonian. You have a professor emeritus in mathematics of Bill Basner, and then you have an award-winning mathematician and population geneticist in Alla Hosjer, and then you have little old me, who's the dumb kid on the block joining the team, and we got published. I'm just like, this is t- t- uh, we're seeing more and more people of high caliber, kind of at the periphery. They're pro ID. They're not really, the peer reviewed publications may not be just like in your face pro ID, but it's slowly getting there. And I had an interview recently. I mean, I conducted an interview with Robert Matheny. He's a cardiothoracic surgeon,
hmm.
and he figured out how to grow heart body parts, specifically heart valves, because he's a heart surgeon.
Right.
And he said, "Look, this is using all this synthetic, like, for lack of a better word, these like plastic things or like raincoat material to
Right.
make synthetic heart parts." He said, "This is terrible. I mean, you do what you can with the material that you have, but so he started investigating how to use a person's own tissue and see if it could be regrown into heart valves. Prior to that, they were trying embryonic stem cells, which were terrible. I mean, you just get some of these really random results. I mean, an embryonic stem cell can differentiate into all sorts of ways. And
Right.
I mean, I'm totally exaggerating here, but it's like, how come you don't get an
Well,
eyeball? You just inject
yeah,
that.
well, that's a good point that Sanford made in his one book that he was working on I don't know, crops or something, and they thought, well, they just randomized mutations, and then you should be able to like do that, and by applying just accelerating mutations and accelerating the selection, why can't you then create new things? And if you have an evolutionist mindset, that should work, and it doesn't. And so that's another good evidence that this is hard. And just if you just randomly change stuff, the odds that they're going to improve are astronomical. And so it doesn't work. And so you have to be thoughtful and use real stuff, biological stuff that works as your template, as opposed to just this trial and error thing that supposedly, created all of life.
Oh, wait, I'm looking for, I just wanted to show you a little bit of our publication. And then you also mentioned that you mentioned the word topoisomerase. Have you seen the Discovery Institute topoisomerase video?
Probably, yeah, yeah. I went down that rabbit hole. That was just fascinating because I think actually it's involved in like every living cell, isn't it?
As far as we know, as far as we know, I mean,
Right. So well, anyway, so it goes. It's pretty pervasive, and it's like that thing is so complicated and had to be there at the very beginning. You're just like, okay, how how in the world do you get there? Um, you know, I mean, there's so many of those things, though. But that's just a great example.
well, the reason I mentioned it, and I'm going to show this on the screen here. We actually got a peer-reviewed paper on topoisomerase plus some other stuff. I don't know if you can see my screen.
Yeah, I can see it. Yep, I can see
Does it
that.
say SciCalc novel
Yep,
approach?
yep.
Oh yeah, this we got published in Oxford University.
Oh, nice.
This is a peer-reviewed paper. And so Joe DeWeese here, he's Mr. Topoisomerase. He studied this enzyme for like 20 years. He's a Discovery Institute fellow, and he's the one who helped when they released the topoisomerase video. It was Joe DeWeese that gave an hour-long talk,
Mm,
that 90-minute talk. And you're just like, this guy's really brilliant, and he is.
right.
I had the privilege of, so here's me, little old me. Kirk Durston is, he was, it was his PhD dissertation that got this started 10 years earlier.
Oh,
And so you could see we had a big team here, and Thomas Townsley, we wouldn't have made it without him.
what do you mean by ranking critical non-proximal interdependencies? What does that mean?
It's pretty complicated to explain. This is what I'm saying. Where ID is now is just so far beyond where we were even 15 years ago.
Yeah.
Because some of the stuff that we're getting into, it is so complicated now. It's no longer single authors being involved. You're having teams of authors. And so we did this topoisomerase. But since you asked, basically what we're able to do is there are important parts of like a machine that, every machine has parts that interact
Mhm.
with other parts. And when we look at the sequence of a protein, it's like, well, what amino acid interacts with another amino acid possibly, or in cooperation? You can't just look at it randomly. And so there is like a pattern in nature, and it's a design pattern. So we have similarities and differences. So if you take a topoisomerase in, like, say, a monkey chimp, human, a plant, a dog, a rabbit. They have some definite similarities and then some differences. And the evolutionists said, well, that's just random mutation and selection, and that's a pattern you'd expect. Well, we found, I'd make the argument that actually, no, those differences are very well designed on two levels. One, they're optimized in general for the organism. We're finding that out. And that's a whole nother very complex topic, especially when we talk about post-translational modifications. And I have to correct something you said. There's a terminology you used about disordered protein. Actually, disordered proteins are actually very complicated. They're actually, that's probably, they shouldn't use that word. They should use the word dynamically reconfigurable, because they're very
doesn't it really just mean it's not, doesn't have a particular fold? It doesn't like correspond to, like, it just like fluctuates like a random string would?
Depending, it may or may not, because when it has post-translational modifications, which are chemical modifications like flipping bits, it will adopt different conformations. The advantage of that for a very complex organism like a human is you could take the same protein and, like a multi-purpose tool, you can reconfigure it and it's going to do a totally different task.
Okay, so like basically it allows it to like be Swiss Army knife and fold, it
Yes.
when it needs to in the right way. I was just
Right.
thinking in terms of the what that antifreeze protein that Behe talks about, where you got this antifreeze protein. It's not complicated because it's disordered. All you need is one hydrophilic head and one hydrophobic head, and boom, it's like, oh, that's conceivable that natural forces could do that.
Well, in this case, it looks like it's very well orchestrated.
Yeah.
And I had helped Joe DeWeese publish on the issue post-translational modifications in topoisomerase. And if you look at this, it looks like a computer punch card. It's unbelievable.
Yeah.
All these bits flipping on and off, and it's
right.
The topoisomerase, let's say, with 60 different post-translational modifications. Let's even make it 20.
Right. Yeah.
That's like 3 to the 4 to the 20th power possible conformations.
Right.
We cannot possibly. I mean, this is just one protein, and it has all these possible conformations and states. And
you mean different states within a particular organism or across organisms?
Within one organism.
Oh, within.
And so when we look at the roles of topoisomerase, we're like, oh, it can do that, it could do that, it could do.
Yeah, so it has to have an RNA that like toggles that switch.
No, we actually don't know what toggles the switch.
Okay, so well, it's got to have something that's there, and it's following it, and that's a lot of.
So there is. Just get this: you have not only alternative splicing, but once the alternative splice makes a protein, even that protein can be modified in real time.
Yeah.
Not just edited. So the sequence is the same, but you have these ornaments. Just think of it like a Christmas tree. You could put different ornaments on it.
Yeah.
And very complex organisms like humans, their proteins are heavily ornamented. And the ornaments change the meaning and operation, and it's very important to the way it signals. Like I'll give you a very good example. The histone protein that helps form the the DNA winds around these spools called histones, and it looks like the old core memory of long ago in computer science. It really does. It's amazing. And so you have the old magnetic core memory. So you have these spools, but the spools have tails, and those tails it forms a Turing complete machine, and you can flip the bits. So you put these ornaments, which we call post-translational modifications. They're chemical. They put little molecules on the protein. They add it just like an ornament, and that ornament changes its meaning and behavior. So, for example, on the histone 3 in the Hox cluster of DNA, so the DNA winds around these histones in the Hox cluster, a machine called the polycomb repression complex, which is like a read-write head. It's a write head. It'll go to that histone Now remember, there are 3 billion base pairs, and it's going to find that location. It's going to find that individual histone out of 8. It's going to go to the 27th amino acid, and it's going to put that ornament on there. That causes the difference in the quality of the skin between the skin on your feet, at the soles of your feet versus your eyelid.
Wow.
And this is how you get all this. This is one of the mechanisms, one of the many mechanisms.
Is that related to like methylation and epigenetics? Is that like
it's
would you
very
say that's
similar to that. So methylation, you can methylate DNA, but you can also methylate proteins, and that's one post-translational modification.
oh, right.
And this is where I just laugh. I said, we're so focused on, literature like say in Edge of Evolution. It was great for that time, but we've moved so far in advance. We have more examples than we know what to do with. I could just say, okay, why don't
The
you?
thing is that the evolutionists would come back and just say, listen, this just shows that there's so many different possibilities, and those are specific ones, but there's a large number that could happen. So as we get these random changes, everything is, what do they call it, promiscuous, and it has, it just has all these, there's lots of different ways you can have, the histones wind up. So why not just randomly do it? And
I'll tell you the counter argument to this, and I've worked very hard at this, and I wish the ID proponents would move this forward. Okay, there are an infinite number of ways to make a lock and key set.
Right.
Infinite number of ways, infinite. You can't just say, oh, since there's infinite number of ways, it's easy, because you cannot make a random lock and then a random key and expect them to fit. You have to have a preconceptions. I said that a lot of protein functionality involves more than one component. So the earlier ID arguments of, say, 24 years ago, it's like, oh, we got a functional beta-lactamase, which is just a single protein in isolation, and it can do its thing by itself in the sea of chemicals. But a lot of protein systems, the really amazing ones, it's just like a nut and a bolt. All these pieces actually have to physically, so you saw it when you mentioned the ATP synthase. A lot of those pieces maybe in isolation would be totally non-functional, but in an assembly they work. But the other thing is they have to be geometrically able to fit. And I said, you just can't take arbitrary shapes and expect them to connect in a way that will create these amazing functions like an ATP synthase.
Right. That's why I found so frustrating the debate with, like, Axe was saying, he was floating around numbers for his beta lactamase of what, 10 to the 77th, and then the Stozak crew was saying, they were looking at more like 10 to the 12th. So so you have this debate where they're off by a factor of 10 to the 60th, and they just kind of like walk away like, okay, we'll leave it. And I'm like, if you guys can't, there's something wrong with this subject. If these guys are happy, the evolutionists are happy with, people agreeing to disagree on 10 to the 60th, because that's such a big number. It's like, well, 10 to the 12th is big enough, because, add that to everything else, that's going to put a edge of evolution. But they have to concede if it's 10 to the 40th, game over. But, the ligand for ATP synthase that Stozak looked at was obviously one of the, easiest ones to get, because, it's one of the most common things in your cell. So of course it has, one of the lowest ones. But just to say, on average, if you make a protein complex and it needs to link up to something else, it has to form, and B was very good at showing, explaining this to me, it's got to have, obviously it's got to fit together, and then it's got to have the right, geometry, but it also has the right charge, and to get all that right, that's really improbable. And to show it in a little cartoon of like a half semicircle going into like a little open crescent, like, no, it's not like that at all, because obviously that's very probable that the semicircle fits into the crescent opening. But in real life, it is like a true lock and key. And so then what are those probabilities? Are they on the order of 10 to the 10th, or are they more like 10 to the 70th? And I think that's the kind of problem that we can actually make progress on objectively and hopefully get some better answers to.
Oh,
These are convincing.
yeah, we're getting there. And the first thing is, I personally don't cite Doug Axe's work, not because it was bad work. It's just that we've moved so far forward because that was what we call a monomeric protein. It's just a protein in isolation. A lot of them, like the topoisomerase, is multimeric or polymeric. It means you make two copies of it. The copies have to fit together to work as a complex. And right there is that's a very easy lock and key. And I ran this through I had the X-ray crystallography data on this, and I ran it through a computation, not my computation, something that's used in the industry. There's about 60 connections. And I'm like, okay.
Oh, I forgot. So topoisomerase, is it like the flagellum where you got like 20 proteins working together?
No, the topoisomerase is it's what we call a homodimer. So what happens is you make two copies of the topoisomerase and then do two copies. So like a pair of scissors.
Right.
Actually, the topoisomerase, so you could take, you could say these are identical, but then when you stick them together, they act as scissors. But in the case of topoisomerase, instead of one connection, there are like 60 connections between the two halves. And then it literally does act like a scissor.
But is it one protein?
Yes, one protein, but two copies of that one protein.
Okay, so it's two proteins. It's a protein that needs, like a scissors, those are two, you could say that each blade of the scissors is a scissor thing, but it has to fit together
okay, just the formal terminology, we would say that it creates one polypeptide. The two polypeptides form a protein complex, and we would say the quaternary structure is a homodimer. I mean, they have all their terminologies for this, but just for any of the viewers that are being really persnickety, it's like, okay, I understand this, and I'm just trying to say, sometimes just we use colloquial terms. And so we could actually call that complex of two polypeptides as a single protein.
Right, but wouldn't they just throw the old rejoinder of, listen, we they keep talking about how they, an RNA could make itself, and they have this one template, and then they show how, well, if you create this two other pieces of that same thing, it can form a template to it. So they would say, listen, a long time ago, this one protein was made, and then through trial and error, it formed a template to the other one, and when they fit perfectly, then they were off to the races, and there you go.
So Rob Stadler did a good job of refuting that, but there's some other things. People forget there was an RNA experiment called Spiegelman's monster. And this is something I really want to harp on, because this is when I said, from the days of Eve and Black Box, Darwin's Black Box, and Edge of Evolution, he was kind of he was like the invasion team that, formed the beachhead. But since then, there's been a flood of scientists generations later that grew up on Behe, and they've taken the argument so much farther. And actually, the modern arguments are actually easier to make. And so let me give you, this is the nature of some of the work I'm trying to do. I'm trying to get, I have a delayed enrollment. I'm in a PhD program in biomolecular engineering, and I do want to get into this. I was a scientist and engineer in the aerospace and defense industry, and then John Sanford made me a biologist, and now I'm kind of combining the two. But anyway, so the first thing is Saul Spiegelman's experiment. What they call natural selection, and even though Randy Galuza and I of the ICR have differing views on how to prosecute the ID argument, we both agree natural selection is probably the wrong word, because it's neither natural nor is it really selection in the sense of goal-oriented. The most accurate description is what Darwin's theory describes is elimination of the least reproductively efficient.
Okay.
You're really eliminating the parents that are not very, you're eliminating their line from the population. And when you look at it that way, it's like, okay, you're going to remove the least efficient, reproductively speaking, going to remove the least efficient, reproductively speaking. So in Saul Spiegelman's experiment, and you can see this on Wikipedia, they're playing with this RNA and it was replicating and replicating. They started out with, before I get this messed up, why don't I, Spiegelman's monster, Spiegelman's monster. Before I mess this up, I'm going to put this, since it is on Wikipedia, this is hilarious because this is played out in so many ways. This is a simplistic example, but this is when they began. This is the beginning of bad news, and it has been since verified on many levels. Okay, Spiegelman introduced RNA from a simple bacteriophage, Q beta, into a solution which contained Q beta's RNA replicase, some free nucleotides, and some salts. In this environment, the RNA started to be replicated. After a while, Spiegelman took some RNA and moved it to another tube with fresh solution. The process was repeated. Shorter RNA chains were able to be replicated faster. So the RNA became shorter and shorter as selection favored speed. Over 74 generations, the original strand with 4,500 nucleotide bases ended up as a dwarf genome with only 218 bases.
Right. Yeah, that gets into genetic entropy, right there.
Yeah, and so, when I first put this, before they let me get to the punchline, they said, "Oh, Sal, that's so irrelevant." And I'm just like, "No, it's not, because let me show you some more." This is Lenski's experiment. Let me see. This was, hang on.
Yeah.
Oh my goodness, this was.
Sadler does a good job of like highlighting how Lenski's experiments show that, we get these trivial changes and we've done it over, I don't know how many generations, 60,000, 100,000. And you get these trivial changes. They're generally degradative.
Okay, so you have genomes decay despite sustain. Okay, this is what's so funny. If I see if I could expand this. Lenski is like, is Richard Lenski kind of embarrassed to have his name? So he sticks it in the middle so you won't notice. I'm just kidding here.
Yeah.
But look at this. The genome, so fitness is increasing. So just like Spiegelman's monster, it's replicating faster, more efficiently, but the genome is actually decaying. Now, I didn't say that. It is Mr. Richard Lenski himself, and this is one of the strains in his infamous long-term evolution experiment. I showed this to Dr. Sanford. He just started laughing. I said, "Hey, John," I said, "look at all the work we've done. It's just summarized in one phrase."
Yeah, it's very telling that he retreated to a digital replication over the biological as like a fruitful way to make his point, because like, listen, the science is all about repeatable experiments and with real stuff, not things with assumptions. And if you look at fruit flies, if you look at bacteria like Lenski's experiments, if you look at what's going on with malarial and anti-malarial drug resistance, those are real experiments, and they don't show the power of evolution. They show very trivial changes, and they show degradative changes, as you show with the Lenski's bacteria. And I think the rejoinder to that, Coyne mentioned that Jerry Coyne mentioned that, well, that's just because it's artificial. If it was in the wild, then they'd be fit as we normally experience it. And it's like, okay.
Well, but the problem now is they're actually because genome sequencing is cheap now.
Yeah.
They could say that decades ago, but we can actually test this now, and it's embarrassing. It's embarrassing. So let me just, this is also a very respected peer-reviewed journal. Look at the title.
Right. Yeah. And I'm sure they don't want the takeaway to be that evolution is impossible. They want it to be that, well,
let's see.
Curious.
Have you heard the word reductive evolution?
No.
Okay. Hang on. I'm doing a dominant
But
Oh, okay. So reductive evolution means the genome gets reduced, meaning it loses genes.
okay.
So like Spiegelman's. All
Right.
right. Probably the top evolutionary biologist in the world, in my opinion, is Eugene Koonin. He has a staff of 30 evolutionary biologists working for him at the National Institutes of Health.
Is he still
One of,
going? He's been around a long time. Is he still publishing?
oh yeah,
Okay.
he's probably up to 500, 600 publications by now. And that's because he has a huge staff. He's the head
Yeah.
guy. His name gets on the publication. He has this. I mean, that this is customary. You know, it's his lab. He gets
Right.
recognition. He may not be what they call the lead author. He'll usually be the senior author, or you
Right. Right.
know,
Right.
the senior author.
Yeah.
And I actually got to study under one of his staff, one of his evolutionary biologists at the
Oh,
NIH. And but so reductive evolution is loss of genes or
right.
loss of DNA
Function.
at least. Okay, if not genes, it's now this is a 2013 paper, and I'm just like, guys, if you think I'm cherry picking, genome reduction is the dominant mode of evolution.
Yeah. Yeah.
And here's another one. Let's
Yeah.
see.
Yeah.
Um,
Yeah,
I,
and I think several ID guys have made the point that it's true. Evolution works at survival of the fittest. If you have a dwarf zebra with a bad hoof, he's going to get selected out, and that's going to keep those things out, and that's going to be very constant. So yeah, I mean
so,
that's a good point, but it's not going to make the new protein machines that we see.
so what I was saying is, I said, look, if this is like going to the casino, you might find some exceptional cases where someone happens to have a very lucky day at the casino, unless he was a card counter like me. Um, I got kicked out of the casino for
Oh,
card
there you
counter.
go.
But let's say your random slot machine player. There are some that are very skilled. They identify the machines that are vulnerable. They're called piggy bank.
Hmm.
Uh, that's a whole nother story.
Okay.
But okay, let's call it. How about the roulette table? So
Right.
in general, it's very hard to influence the outcome of roulette. In fact, they work very hard to make sure it is as randomized as possible.
Sure.
And the expected value is a loss of about 5% against the the casino patron. So 5% advantage by the casino, what they call the house.
Right.
And so you might get some unlucky every now and then, but the law of large numbers will dictate over time. This is going to be a losing proposition. So if the dominant mode, if most of our observations, experiments are
Yeah.
showing the dominant mode is gene loss, I said, look, you can't be counting on millions of years, because it's not even taking, it didn't even take Lenski more than a few decades to wipe out 5% of the genome that he
Right.
was. So he'll brag he got a new
Yeah.
function. It's like, well, one, you didn't create any new, what we call truly novel genes. You ended up wiping out promoter regions,
Right.
so control circuits, and you're bragging. So yeah, you got new function, but it's like, well, yeah, because you kind of knocked off the control circuits. And of course, it's not just expressing away. It wouldn't be able to compete in the wild because it's not, you're having to coddle this thing because it's consuming resources. But this is totally, this is nothing to brag about. But I said, if you lost 5% of genes in only a decade, why do you think you're going to have a million years to build this? It's so easy to destroy them. And what we found out under strong competitive selection, what I said, this is like a hiker. He said, well, I'm going to optimize my hiking for speed. And so he just dumps all his winter gear, his
Yeah.
provision.
That's the main
And
argument I want to be's books, right? One of is like that that the loss of if you're running away from the cops, the best thing to do is to break your rear view mirror and stuff, and or if you want to like get better mileage, you'd break you'd break the rear view mirror, stuff like that, because it'd be simple, and it would obviously increase your mileage because it'd be less drag. But and usually the first order effects are all stuff like that. It's breaking stuff, and that's what Lenski found, right? That you break some stuff, because if I'm a bacteria and I've just got this one food, and my optimal thing to do is just junk everything else that I don't need, and there you go. And it's
So what really changed is, so they're giving, the early rounds of Lenski's experiment a lot of fanfare. We criticized, and then there was backlash against the ID movement. But then there was Scott Minnick and Dustin von Hoffagen and another professor, I can't pronounce her name.
yeah,
Just
that was great. That was a great thread.
and,
When he
and
showed how trivial it was to get that what he thought was like a speciation event, and then he realized, no, I can do this in like 2 days if I select for it.
yeah, and then they
Because it's
showed all
really a
these
trivial number. Yeah.
yeah, there are these piles of petri dishes, and Lenski said, "Look at our accomplishment." And Dustin had a little pile like that. Said, "Look, I did the same with just these many." And so, the important thing about that is we had someone who is on our side. He not only did he publish, but he got someone from the National Academy of Sciences to say this was correct.
Yeah.
That and it was really kind of a slap in the face to say, Lenski, you had no right to be hyping this. He didn't,
Yeah.
The reviewer at the National Academy didn't go quite that far, but the ID proponents are like, well, yeah, this is still a big slap in the face. It's like, hey, you had all these papers, you're getting all this fanfare and celebrity status. You found basically nothing.
Yeah. Yeah.
Nothing that was worthy of all the fanfare.
Right.
And then Scott Minnick, especially Dustin von Hoffagen, they studied so many of the genes in that. And he said, well, Dr. Minnick said, look, they lost a lot of the glucose genes. So now it
Right.
can't live on glucose, because
Right.
what was happening is you want those genes if you're in a glucose environment, but they're in the citrate environment or whatever environment. And it's like, well, this is like spare tires in that environment. And so you're dumping all these things that would actually be really good. And he said, you're constantly losing versatility, just like the hiker, just dumping all this stuff. I'm just like, okay, now you're just ill-equipped to deal with a variety of environments. You had all this great equipment. And I said, a good deal, natural selection. So this is where I was saying that this is not going to stop. We keep getting more of this, and
Right. That's what makes it fun. Is, is you learn about these, because I think it's true. It's the more information we get, the more confirmation we get that it works, which is very unlike, say, my experience with macroeconomics, where the more information we got there was like, oh, we thought we'd solved the latest model, and then we get a new regime, and the next business cycle would show that our prior models didn't work because they didn't predict that one. And then we'd fix everything and predict all the last business cycles, and then the next one would come and show us, oh, well, it doesn't work again. And of course, you would only get data once every 8 years or so. But after like 3 or 4 cycles of that, you just got to go, this isn't looking good. How long do you want to keep doing this? But with this ID stuff, it's like the more and more information we get, the more we get these like silly things like Lenski, making a silly, over extrapolation and interpretation, and then he gets refuted, and then he says, well, I didn't really mean it that way, but it's at a high level, it just shows what we were saying. And let's just move on to something else. It's sort of like what Wells's Icons of Evolution, where they say, well, that's that. We didn't really mean that. That was just meant to be, something to show that, or Dawkins' Weasel program, oh, yeah, that doesn't really work, but it works in spirit, and that's really what happens. And you press them on it. It's what Scott Alexander calls the Motte-Bailey argument, where you make this, indefensible claim, and then when someone checks you on it, you say, well, I didn't really mean that. You retreat the Bailey and say, I didn't really mean that. That was that was a simplification for this general idea that's true. And then you're like, okay, so they're just throwing a big spread argument where every time they say that here's all this proof of evolution, then you say, okay, well, let's look at this one. And you look at it, like homology proves evolution. And you look at it, and then you find out, oh, that doesn't work. When you look at all the convergence and all the supposed horizontal gene transfer that's going on, that doesn't work. Like, okay, that's fine. But we've got this other argument. And then, there's an infinite number of them. I'm sure in my lifetime, you're never going to convince the evolutionists they're wrong, but at least it'll make it more legitimate, move Overton windows so that, people like me and won't feel ashamed for believing in creation, because it's a legitimate hypothesis. It's a defensible hypothesis. Like before I read Ben Behe's book, I thought it was patently indefensible. It was like a stupid argument. And so that made me just shut myself off to the idea that we were created, and it shut myself off to thinking that if we were created, we had a creator, and that creator is like a god. So I didn't believe in God at all. But by just showing hey, we're not going to prove it to the world, but we can show young people, or people who don't have an opinion on this, it's a defensible opinion, and you don't have to be embarrassed. Because like, I think Eugene Koonin is a he's an evolutionist, but he's a multiverse guy. So it's like, okay, you believe in a multiverse, and you say that believing in God is believing in a miracle, and you don't like miracles. Like, a multiverse is pretty miraculous. So pick your miracle.
I wanted to acquaint you with some. Okay, here there. Before I forget, have you ever heard of Aaron Ra? Aaron Ra?
Yeah, he's a Satanist guy, like he tries to make himself look like Satan.
Yeah. Okay, so this was so funny. Debated him. I didn't, the debate didn't go well because I had some conflict with my debate partner, and I told the host, I said, I don't want any more partners.
Yeah.
But anyway, the debate partner was Cindy Lincoln, and she got Aaron Ra to have a little bit of a dialogue over the emails. And in the debate, I said, all major protein families do not have a common ancestor. And unfortunately, I don't have the slides. And they all kind of the opponents scratched their heads. And then they came back to me, and Aaron Ra was pretending like I didn't even say it. He said, there's no effing common ancestor for all proteins. And I was like, well, thank you very much. You figured that out. And actually it turned out there was Ford Doolittle, I think. He was saying, well, we hope when we get one functional protein, then all of them can descend from that. But that doesn't look like the way it could be. And I'm like, yeah, it's so obvious. It's so obvious now. And I've asked evolutionary biologists, do you think all major protein families descended from a common ancestor? They'll say, no, they don't. But we all descended from the same organism. But even though I'm just like, okay, I don't think you're really connecting the dots here. There's a problem. Right. It's like, even if you accept universal common ancestry at the organismal level, all these proteins I said, okay, you have a topoisomerase and a collagen and a zinc finger. And I showed them these pictures where I colorized. And I said, you could even visually see that it's impossible. And unfortunately, I don't have that handy, but I've let me see if I could call this up on my screen. But I just wanted to tell you, it's gotten so easy. And I'm just like, do you not realize you just gave away the store when you're saying, because you have no explanation then. Okay, so I'm going to see if I could find this here. I have this on my YouTube channel. Hang on, let me just kill the audio here. And I'll show you this on the YouTube channel because I made a video just specifically on this. And okay,
really just a generalization of the idea that like all the major phyla, you don't have any like progenitors, parents to any of them.
Well, the thing is it is so blatantly obvious they don't even challenge me on this anymore. So this is a spelling. So this is a collagen protein here.
Yep.
So you're familiar with the amino. Okay, just for the benefit of the readers, I know you're probably familiar with this, but for the benefit of the readers, let me see if I can. So there's collagen.
Right.
It's a beauty product, but it's actually something that's about a third of our proteins by weight are collagens. Mm-hmm. And we have all these amino acids. We can label them with English alphabetic letters, and they all have various properties you can Venn diagram. And so the collagen is spelled like this.
Okay.
But there is a non-random pattern here. And I'll colorize it. It's every G, every glycine amino acid, or sometimes we use the term residue.
Wait, what are
There.
the rows? The rows are, is this like a, is this like 1,000 aminos in this guy, or is every row a protein?
Every row, I mean, this is just a wraparound. It
Oh,
would be
right. Okay.
linear.
Okay.
But you actually see it's actually provident that it has to wrap around, because it's in the wraparound, you can actually see a
Right.
non-random pattern. That's a collagen.
Right.
Every third letter is a G. That's very important for the structural properties.
Uh-huh. Right.
And then there's also another so that's kind of the motif, G, XX, G,
Right.
XX, G.
Right.
Now there is another protein called a zinc finger.
Okay.
So if you look at the zinc finger like that, and it's
Wow.
spelled, it only has 500, there's
Wow.
also a non-random pattern
Sure.
like that.
Right.
And by the way, see, this is where the zinc ion would connect to the C. There's a C here, cysteine, cysteine, histidine, histidine. And each row will have a zinc ion on it. It's very important for the way it functions. So you can clearly see a collagen and a zinc finger are not the same. And I said, okay, so I'm going to force the issue. I said, there's no way this has a common ancestor, because a collagen needs to have this architecture to function. If you change it, it's going to cease to work. Same for the zinc finger. So I colorized some of the other columns here in the zinc finger. And so now they won't even argue. I said, okay,
Right.
so now you've got a problem. Where do these major families come from? Not only are they sequenced different, I can even demonstrate that visually. You can demonstrate it computationally, but even in terms of their shape. So
Right.
if I showed you all the different shapes of proteins, um, uh,
Yeah, they're crazily complex, and uh I like the one visual of the like the creation of the flagella, because they show how all these proteins get interlaced, and they work together in this little, they look like they're made like a rope cord, and that the way they're built, um and it just, it's the way you would make it if you were an engineer.
oh, so the argument, and I mean, if I may just self-promote a little bit or a lot. I mean, it took me a while to think about this. Let me see if I could I have this on my YouTube channel. I'll send you a link to my YouTube channel if you're at all remotely interested. I'll send you links to some of my, um,
But when they
uh,
say, "Oh, okay, there's some progenitor, and it happened upon like the little zinc repeat, and it happened along the collagen repeat, and then once that worked, then it just went to the races and went off down that rabbit trail, and then the zinc guy went down his, and the progenitor we don't know, but who knows, millions and millions of years ago, we don't have the data, but and so you're just like, okay,
well, the thing is they've gotten to the point where they don't even, they say no, they don't have a common ancestor. And then I'll ask, okay, so describe, say, a scientific theory. You say this is the starting state,
yeah,
this is the end state, and then you describe all the processes where you get from start to finish, and it's just all blanks. I said, one, you only know the end state, you don't know the
right.
start state, and you don't know the process that got you there. I said, how is that a scientific theory?
Yeah, but that's the key, isn't it? That the fitness landscape is not like the modern synthesis showed, which was this nice, smooth set of functions and these hills you climbed up, and instead it's just you got these spikes that are in the middle of a huge cliff. It's just like a big spike up, and then yeah, you can change several. I don't know, like I think Brian Miller might have estimated, like, or Max might have talked about, like, on your average, let's say 300 amino protein, like what's the average number of aminos that your average protein will tolerate? Do you have any sense of that? Is it something like 10%
Oh,
on
it can be
average?
it can be up to 90%. And that's another thing. It depends on the protein.
Right.
So like the beta lactamases, I've seen them, and just like you could modify about 95% and it would still probably be a beta lactamase, but then it's not optimized for the organism. The figures that I saw, they call it the twilight of um the twilight zone of protein
Right.
identification, and they said below about 12% similarity, you won't be able to identify them as.
And they won't work on average. Yeah. I mean,
So,
obviously, like you
so
said,
some proteins can tolerate a lot of change. So the
right.
way I liken this, I said, look, take any random car. There are parts you could totally
Sure.
dispense with, you can modify, but then you have like the critical parts. You cut the battery cable, it's dead. You
Right.
start pulling you pull a couple spark plugs, it's probably
Right.
just as good as dead. And I
Right.
said, this whole thing of it used to be like, say, 60 years ago, the creationists said every amino acid's critical. It's like, no, that's not true. But you can take about a core. So like, let's say you have the topoisomerase protein, which is 1600 amino acids, 1700. Probably at least 170 are just like really, really critical. Joe DeWeese, who's been he's Dr. Topoisomerase. In his dissertation work, he found 2 amino acids. He said any one of these two, if you knock it out, the whole thing's gone. Gone totally.
Okay. Yeah.
There are probably other parts of the topoisomerase. If you knock it out, it's gone. Just like it's just super critical.
Right. I think they were talking about, this gets back to the Winstar, where they said, hey, cytochrome C varies across organisms, or I don't know what they were looking at, but it was something like that. And it's like, okay, it's a very complex structure to work. And then someone said, the probability of getting this, is infinitesimal. And then the rejoinder was, well, yeah, but different organisms have different cytochrome Cs. So there's dozens, maybe hundreds, thousands that work. And it's like, okay, let's say there's 1,000 that work. Well, what's the state space of possible, cytochrome Cs? It's 10 to the, 100. So 1,000 divided by 10 to the 100 is still, it's 1 in a, 10 to the 97. So, who cares? That's impossible. But they would just focus on, I've had debates with this. People said, there's lots of different ways, you can get there. It's like, okay, lots. You're saying there's 7. That was the thing in the Behe debate with about the antibacterial resistance. He says, there's several ways to get it. It's like several. Okay, several doesn't matter. We're talking about odds. We're arguing about odds of 10 to the 20th. If you want to multiply that by 7, go ahead. It doesn't change the debate at all. It's just like, that's not relevant. That's not going to save the problem.
Okay, so if you saw me just kind of panicked here trying to, because I'm looking for stuff to show you.
Right.
And a lot of it's on my web, out on the web.
Right.
Okay, so we can kind of build a phylogeny of cars, like the first one was, first automobile was by Otto himself, Carl Benz, I guess of Mercedes-Benz fame.
Probably.
And then all of them kind of have the same, have some of the same,
wheels, internal combustion.
By the way, I think this is Danica Patrick's Formula One, and I also love dragsters.
Okay.
But there is a problem. You cannot do that for the parts. That is, there's no one part that will descend into be all the other parts, even just in terms of geometry.
Right.
A tire will not have the same ancestor as this is a gas tank or a piston. And I said, it's obvious that this carries over. This is why, and I have an evolutionary biologist on record. He said there's no common ancestor for all proteins. And so let me just show you some of the, here is a good picture of some of the proteins. So on the left, this is zinc finger. This is a homotetrameric potassium ion channel. This is a homohexameric helicase. This is a homodimeric topoisomerase, a heterotrimeric collagen, and a homodimeric insulin receptor. I mean, they're shaped so differently. Not only so I showed you the zinc finger sequence and the collagen, they're spelled differently.
Right.
Not only that, they're so radically different in shape. And I'm like, there's no way this has a common ancestor. I'm just kind of beating a dead horse here. And I'm just like, okay, so now you can't, how are you going to invoke evolution from a common ancestor? So I asked Mr. Aaron Ra, I said, so can you tell me what the ancestor looked like? No, you can't. So can you tell me the process that went from this ancestor you can't describe? And it's like, okay, now you have a process you can't describe. So you have a process you can't describe, you have an ancestor you can't describe, and you're claiming it's true. I said, that's just not going to fly if you're going to claim. I said, look, I'm a religious guy. You can claim it as a faith statement on your part, but don't try to pass it off as anything like a real scientific
Well, I
theory.
know, it's funny that they keep, criticizing ID for God of the gaps when it's always like one can imagine. It's like, well, one can imagine, that, yeah, you can imagine it. I can't stop you. But if you just look at the basic aquaporin, which you need for the first cell, those things are so incredibly precise and well specified, and you're just thinking, okay, how how did you get there? Because talk about a lock and key. Those and aquaporins are like the protein complexes, right? And they fit together, and I think they strip off the ion from every water molecule that goes in and out, or every extra electron or something. They're just so precise, and it's like, okay, what was the progenitor to the aquaporin? Was it, how did you, like, so the original aquaporin, it was just a big sieve, and then all of a sudden it just decided to get so precise it could strip off a single electron off of a water molecule. Like, really? That's step by step. Like, what would select for that? I mean, because the
Well,
state
yeah.
space they'd have to navigate was just insane.
Well, we'll see. Then it's even worse than that because it can't even. Not only can it not select, because under strong selection, Linsky's experiments and numerous others have shown it can't even do that because it's destroying genes faster than it can really build really novel functions. So it's like they were just giving this illusion that it can be very constructive, whereas it's just dispensing stuff. I mean, it's gone. And in a bacteria, these microbial where there's horizontal gene transfer, there's chance of recovery, but not for complex organisms,
Right,
multicellular. Once it loses the gene, it's probably good as gone forever.
right, right,
Um,
right.
and so I did want to show you this. So we're talking. So there is a testable prediction that even outside of ID, but it supports the ID hypothesis, particularly genetic entropy. If the general trend is toward gene loss or disruption, then there's going to be, um, even with selection, there's going to be general destruction of genomes. That's a testable prediction. And I said, we're going to find out. So I was in a debate with an evolutionary biologist, and I asked him, I said, can you name one major population geneticist of good reputation that thinks the human genome's improving? He
Right,
couldn't name one. He
right.
came out to debate me about genetic entropy. I said, just name one guy that thinks we're improving. Couldn't name one. I said, look, I felt if anyone were really paying attention, it's like, I think I just made my point, that whole debate. I said, name one guy on your side. I'm not asking you to name a creationist. Name one guy that you respect that thinks the human genome is improving. And he just, he gave a non-answer. I just like, okay, I think
Yeah, that
anyone
gets
that's
into
really,
that whole punctuated equilibrium stuff where they say, okay, we degrade, and then you get what is it, Goldschmidt's monster or something, where you just have a boom, it just stumbles upon this great, you know, new configuration, and then it goes to the races. Like, okay,
so
could
I'll
happen,
show you this paper and intelligence and childlessness.
right?
Okay,
Yeah,
this wasn't done by a creationist.
right.
Um, um,
Yeah,
More intelligent men and women are likely to desire childlessness. More intelligent women, but not men, are more likely to become childless.
right.
Due to just genetic fertility, the average level of intelligence is likely to decline.
Yeah,
And I'm like, oh, so again, natural selection is eliminating the least reproductive, the least prolific, the least reproductively efficient. And so you have very intelligent women that end up not having kids. And I'm like,
yeah.
okay, how does this process again start to develop really fabulous brains and the intelligence we've attained? Um, and
Yeah.
and
Well,
and
that gets
just like, okay,
yeah, yeah. I think they revert to the punctuated equilibrium thing where, yeah, okay, on average it's, there's entropy going on, and we get whatever it is, 100, 150 mutations in the genome every generation. But, there are these saltation events where, every so often, the herd just gets wiped out. And then, the Superman, he gets, replicated a million times.
Okay,
Well,
I mean, occasionally there are these things just make me laugh. This is what evolutionary biologists do. Were there
right.
evolutionary advantages to premenstrual syndrome?
Yeah, right,
And then,
right. Yeah.
um,
Yeah, could
and
be.
then I don't
Yeah.
know if you saw this.
Mm.
I shared that this is a New York Times bestseller. This is hilarious. Um, this is such a hilarious title. Um, because they have such a scarcity of examples of natural selection building something, they
Right,
do have an abundance of birth defects that
right.
they cite. And this is a New York Times bestseller. I shared it with Dr. Sandford. He laughed so hard.
Oh,
Survival of the sickest. Um,
oh,
a medical maverick discovers why we need disease.
oh. It helps us fight off viruses or something, or
Like malaria,
yeah, right, right,
sickle
right, right.
cell. And I'm
Yeah,
like, okay,
yeah,
I
yeah,
said, you guys are making my job as a defender of ID so
yeah,
easy these days. I mean, I don't have to work very hard.
right. Well,
Um,
it's interesting that the evolutionary argument. I think it is important to recognize that every biological process or species has to have it has to be an evolutionarily sustainable um configuration or force. So like, if it has to be, it's fit in the sense that, it has to be efficient. So, but that doesn't, for a lot of evolutionists, they'll say, well, that shows how evolution works. But I'm just like, no, obviously, almost everything is evolutionarily sustainable. It's fit, but that doesn't mean that it rose, through this hill climbing process in our evolution. Because yeah, the state space to get to where we are is too spiky. It's too perilous. But it is true that, you know like, just like, when we look at things like virtue and like, what are good, virtues? Why is faith, hope, and love, why are those such great foundational values? And it's because it's a evolutionarily sustainable ethic that promotes human flourishing. It promotes, happiness. It promotes things like that, and it has to be. I mean, it can't be like the Shakers who say, "Oh, well, we don't believe in procreation." Well, Shakers aren't going to be around long, and they weren't. So it is great. That's one of the things that turned me on to Christianity was like the not just the principles, but the prioritization of the principles was just so consistent with, human flourishing and happiness. I just thought, well, that can't be an accident. Like this guy stumbled onto this 2,000 years ago when, supposedly, everyone was an idiot. How did he figure it out so long ago? And all those. So it just amazed me. I mean, that's what pushed me over the edge from deism to Christianity. It just the the prioritization of principles and just seemed just was too good to be true. I mean, if you look at anything else like Stoicism, it just doesn't work. It leads to nihilism. Like, the virtues are good, not the person, and so it would lead to meaningless and nihilism and despair. Whereas Christianity, it leads, I mean, a life without faith, hope, and love is, the opposite of hope is hopelessness, and that's terrible. And what's greater than love? I mean, that is the most important part of the stool. Like, wow, does anyone say that in the Stoic philosophies or the pagan philosophies? No. Um, the thing is, they would come back and just say, well, every 10 years they fix them, but when they find out that they're, the prior, progressive or humanist, would I think they've had three sets of humanist principles. They change every 30 years, and they always turn out to be disasters. Because the first two were like, promoted Marxism, which was a total disaster. But the others are just, when you put them into practice, it ends up just being dystopian, and it's like, that should tell you something. Well, how did these old guys in the Bronze Age, figured this all out, and all these smarty pants today, just end up making, these dystopias and all these, depression and nihilism. So, but sustainability is different than the fact that structures are designed to be or have to be sustainable. That doesn't mean that that explains how they got there. It just means that's why they're there.
I have a couple of things before. I mean, this isn't necessarily goodbye. We could probably talk again sometime. There are a couple things. First off, we'll get to your. I'd like to talk a little bit about the anti-weasel work because I'm interested in that. But before I get to that point, you had mentioned that you had come to accept there was a creator.
Yep.
At some point, and how long was it after? Did it take before you went from that to becoming a Christian?
I'd say it was it took me like 10 years to accept that evolution was wrong. And then it took me probably another 6 or 7 years to decide Christianity was the best. Because once I decided we had a creator, then it's like, okay, is it like, Nick Bostrom's simulation? There's like some little kid in sixth dimension. Is it Gnosticism? Is it, the Rastafarians, or, it could be anything. Aliens. And, and so then I just kept looking around, and that took me about 7 years to figure out. And, once I started to really read the Bible, actually, I started with Proverbs and Ecclesiastes, which are the most, those are the most rational parts of the Bible, right? They talk about ethics, and I thought, wow, these are really good ethics, and these are really good ways to look at your life, and very consistent with like Aristotle's whatever he talked about, like was it Nicomachean ethics? It's very much in that strain, but it works a lot better than Aristotle's Nicomachean ethics. But yeah, so it was like 10 years to accept evolution and 7 years to accept God. So I didn't have a road to Damascus moment, unfortunately. But I was one of those rare guys that got there through my head and not my heart. And and it's like I said, the more I started reading about Christianity, I read Proverbs and Ecclesiastes, and then I'm like, oh, these are good ideas. And then I started to, then I did a full Bible study, and then I saw how it all fit together. And it really wasn't a life manual; it was more like a story. And then it made just so much sense. And it just started to all come together. But yeah, that took, it was really helpful to read the whole Bible, start to finish, and understand that. And that took, reading it and understanding the Bible, that takes at least a year, right?
So you mentioned, like the aliens or whatever. I got to tell you, my favorite. People ask me what's my favorite creationist work. I'd say it's Alien Intrusion, the video by Gary Bates.
No, I haven't seen that one.
Dr. Stanford makes an appearance in that video. It makes a very compelling scientific case.
Yeah.
I mean, it touches obviously in the spiritual things, but the first half made a very compelling scientific argument against aliens.
And that's and I've always been a fan of Ancient Aliens, and actually Michael Heiser, who died last year, he's really good on this, and he points out that people who are into Ancient Aliens and all that stuff and ufology, they're primed for learning about Christ, because they're looking for meaning. They're looking for how can I explain what they're looking for, some transcendence, something.
Yes.
To give them, and they're looking, and there are answers that will satisfy many of them, and actually, if you look at the Ancient Aliens, there are lots of things that can be explained within the Bible, because the Bible talks about the Nephilim, and they correspond to the Anunnaki and and all those things, and they have explanations for, like, when they sent down the sons of God and they had all this special knowledge. Like, oh, that's consistent. Like, how did they make the pyramids? Well, we know that they sent down, to one Enoch, which is an apocryphal book, but it was quoted, and it was respected by many early Christian fathers, including it was in Jude and in 2 Peter. So yeah, but anyway, the whole thing just all starts to make sense. And like I said, if it's cool when you read something, and the more you learn, the more it makes sense.
Well, I wanted to tell you that was very, very heartwarming to when you emailed me and you told me of your conversion. It really did lift my spirits, and I said I wanted you to have a chance to kind of share with my audience here, because
Yeah,
it'll.
so if you would have met me 20 years ago, I would have been one of those guys on their side telling you you're a bunch of idiots that don't understand science. And so that's the way I thought then, but it was out of ignorance. And so yeah, that's a case where you can make it you're not there's no chance you'll convince the world. But there are people like me, and I think there are a lot of people like me who, they're just ignorant about like all these basically lies, and Satan does lie, that they're telling us, and they accept them because of peer pressure. I didn't want to look stupid. Most of my friends were atheists, but then you realize that the arguments they have just don't hold up, and so yeah, you always want to be talking to those people. And yeah, Aaron Ra, he's never going to buy its story. Fine, um, but that doesn't matter. You just got to show, hey, they don't have an answer for this. This is an important test. Lay it all out, make it easier.
And so we can continue the conversation some more. This is very pleasant, but how this all began, you were trying to show me some of your work on the anti-weasel. This is a little bit embarrassing. Every one of us approached the ID argument in such our own specialized way that
Right.
I actually have a hard time kind of like when you were quoting the stuff in Edge of Evolution, I was just like, okay,
Right.
I did read parts of that, and some of it
Right.
was just like, okay, this is not how I comprehend the ID argument anymore.
Okay.
I want to say thank you for giving me a chance to show you how I comprehend it. I didn't go into the careful mathematical details that you're describing when you're also citing, Ken Miller and Larry Moran. But what I tried to show today is I said, I think what has happened, we have so much data now that it's just like, well, I would start, the arguments I use don't go back to Edge of Evolution, because
Right.
Behe himself went to his other book was Darwin Devolves,
Right.
which was I just like that. That's an even easier argument to make.
Right. Well, there's an infinite number of ways to prove, there's an infinite number of ways to prove something wrong. There's an infinite, there's a lot of ways to prove something's right, Pythagorean theorem. This is one particular argument, in Edge of Evolution that I thought was interesting, because they were debating about, how, what are the probabilities of getting, two specific, mutations out of 424 amino acids, and B was saying, well, it's the probability of getting one times the probability of getting the other, and so that's basically like the probability of a simultaneous mutation. And they said, we're not going to look at the numbers. What you're saying is obviously stupid, and we don't accept it. And Dawkins and Miller and Coyne and Moran, they all said, on principle, we're not going to discuss it because that's a stupid thing to say. And I'm saying you can mathematically show that as an approximation, the simultaneous mutations is close. It's not true that the indirect paths add up to something qualitatively different. So as just an eyeball, you're within a factor of 2 if you say the probability of getting the right 2 mutations simultaneously happens in one shot, because the probability of getting one followed by the other, since the first one's neutral, well, guess what? You've got all those other 424 aminos, and the probability that one of those, goes the wrong way is much higher than the probability the one you want changing. And it turns out if you just do the math on the probability space, and you can map it with a trinomial lattice, which is really cool because it simplifies things. And you have just a trinomial set of nodes for only 424 proteins. So you don't have to do this Kimura like long-term thing that Behe and Snoke did. But that's just one little point in the argument. There's never going to be a decisive argument in this argument. But the fact that you can mathematically refute this intuition that prominent evolutionists have used, I think that's an addition to the literature. That's what I was trying to argue.
Oh, thank you. And I hope you'll forbear with me. it turns out maybe the reason you actually approached to talk to me, it's actually I could tell as I read it, I said, I cannot add any insight or help you along,
Yeah.
or because it's actually, I mean, I have a math degree and I've studied physics, but to really um, uh, to really, I would have to invest time
Right.
to go
Right.
back and
Yeah.
just
Well,
think
as you
about it
find out in
carefully.
your work, all these arguments are parochial and complicated, and they take an investment of, several hours to just grok, like, what's the setup?
Yeah.
What's at stake? And it's like, you just don't have enough time to go down the rabbit trail of investing. I mean, I spent hours, trying to understand what they were arguing about. Because when Ken Miller said, it doesn't matter what your numbers are, I don't agree with, your philosophy on like this point. And I'm like, wait a sec, what do you mean you don't care what the numbers are? How could you say that? And so then I had to figure out, well, what is his conceptual problem? And his conceptual problem was, no, there's intuitively, there's the number of paths to get someplace, to an indirect target is going to be vastly greater than the probability of simultaneous mutation. Um, and it's like, okay, yeah, the number of paths will be infinitely greater. But the thing is, the state space blows up at an exponentially much greater rate. So you don't get there faster. And actually, if you want to win, you want to double your money in Vegas, the best thing to do is not to go there. If you want to double $100 in Vegas, you don't go there and play, $2 in the slot machine, 50 times in a row. You do is you put $100 down on the, on red, and then, there's a 47% chance you'll double your money. It's not good odds, but it's better odds than if you just did it little by little. And in that
Yes.
sense, simultaneous mutations is the best way to reach your target.
By the way, bravo. That's what I couldn't quite get. What you just said, because I was in the gambling industry, that totally just like, oh, wow, thank you.
Yeah.
Piecemealing it. Exactly. So what the casino wants, this is why they have limits on the tables. They want to use the law of large numbers. They want to
Right.
keep as many trials as going because they know the expected value will eventually prevail. So I want to say,
Yeah.
okay, now I totally get your
Right.
argument.
Right. Yeah. So your odds
So
are, much better if you just,
okay, so
put it
now
on the
let me give
Powerball.
you, yes. So let me give you there's one really easy thing to miss in Dustin von Hofwagen and Scott Minnick's experimental analysis. And I caught it because I happened to be studying this on Lenski's. He said Lenski had he was starting, first of all, one reason that Lenski's experiment took 30,000 or whatever generations and all those petri dishes, 15 years.
Right.
He had a defective bacteria. It had a broken gene, and that gene only had five nucleotides. Nucleotides, five that were missing. And I said, look, that would have been a very nice path. You just repair those five measly ones, and then you're
Right.
just off to the races like Scott Minnick's experiment.
Right.
And I said, any evidence that it ever figured out to repair that? I said, look, you have 99% of the gene there,
Right.
and
Right.
you
Right.
lost five. You lost, okay, so let's just say the average, that'd be 1,200.
Yeah.
So whatever, out of 1,200 nucleotides, you lose
Yeah.
five. That's
Yeah.
not even a percent. You're
Well,
99% of the way there.
yeah.
Did
Yeah.
any selection come along and fix it? No.
Right.
And
Right.
I'm just like,
Well, that's it. The paper I sent you shows a model where you can actually calculate with if you have 99, aminos, and you need to like get five new ones or five, change is specific ones. Odds of doing that are on the order of getting those five in one shot. And we know intuitively that the odds of getting all those five exactly right in one shot are astronomically small. And so that's why it won't occur. And so it's not, so if like it's an order of magnitude, you can just say, what's the odds of getting a simultaneous set of five of those mutations? And you do that and you get, 1 in 10 to the 100th. So it's never going to happen. And if you
Well,
add up all the indirect paths, it's going to be 1 in 10 to the 99. So it's the same thing.
so what was really bad about this? Okay, so there's, you have the amino acids, which is in the protein, and then the nucleotides, which is in the DNA.
Right.
So you basically have, for one position of DNA, you have 4 possibilities, A, C, T, or G.
Right.
So if you have 5, that's 4 to the 5th power. That's not astronomical at all, but it couldn't even get that.
Well, no, it is astronomical because, remember, the probability of one of them changing is, it is going to be, it's got to be 5 out of 999 that you get the mutation in the right place. And then of those, you've got to get the right amino, presumably. Let's look at aminos instead of codons. So then you've got, you're going from one amino and you want to target another one. Well, there's 20 to choose from. So the one's wrong. It's basically got to choose one correctly out of 19 it can choose to. And so then if you just say, okay, it's got to do that. So it's 1 to the 19, 1 over 19 times 1 to the 19th, that to the 5 power. But then it's got to be in those specific, 5. And that the odds of that are going to be on the order of 1 in 10 to the 100th. And you can show that mathematically.
And I probably, you're correct. I misstated the problem because it had to fish that out in all the 4 million nucleotides, but it had to identify the location. But I said, look, if selection were as good as you were saying, it ought to be able to figure out, oh, I need these 5. But what you wrote, which is totally correct, for selection to work in the way Weasel works, it has to have foresight. It has to know in advance, this is what I'm looking for. And when it's partially correct, I'll fix it. And the problem with particularly 5, and I noticed this immediately, I said, Dr. Minnick, that's a frame shift mutation, which means if you when you delete anything that is, if you
1 out of 3, just binks them all over. And
well, what happens is when you have a frame shift, you create all these premature stop codons, all these punctuation marks that said premature termination.
Right.
And I'm like, oh, this is really, really bad. Because the entire gene is instantly disabled.
Right.
It doesn't even express, there's nothing to select. It's like, it's gone. And it's like, look, I mean, an intelligent person will say, oh, look, you just need to put these 5 back in and it's okay.
Remember that great thing when they had that new bacteria that developed the ability to digest a plastic or something, and then they said, well, it's because of a frame shift, and then the evolutionists jumped on it and said, this shows how random mutations give new functionality, and then intuitively you knew they were wrong because it's like, listen, if you do a frame shift across a functional amino acid, there is no way that thing is going to survive. No way. If that's what's driving your process, the numbers are astronomical. And then, when they looked into it, it turned out that no, this was a minor evolutionary change. But the fact that they mathematically had the intuition that it was possible shows that they don't really understand. They don't understand the mathematics of this state space. They're kind of like the people who thought that a bunch of monkeys could type the complete works of Shakespeare. Yeah, it's possible, but the odds are so small, it's impossible. And if you don't understand that, and a lot of people didn't for a long time, you think something's possible when it's not. And so their mathematical intuition is wrong because they have a wrong model. They don't understand the precision of this state space. So just unfortunately, it's a better word for it than information, because information just brings in all this baggage to it. But I just think if you just look at what we know about the precision needed for a protein to work, it doesn't tolerate these frame shift. Wouldn't do it.
Well, I got to tell you about that particular one, Dr. Sanford. Okay, Dennis Venema said this is his favorite example, the nylon eating bacteria, which you're alluding to. It eats nylon. He said, this is my favorite example of evolution. And he said he's using it to assail Stephen Meyer. Well, Dr. Sanford called me up. He said, Sal, will you look into this? He gave me some pointers. Go into the NIH databases. And I did. And at first the search was a little weak, but then I got better at it because I got trained by one of Eugene Koonen's
Right.
staff members. And so the first thing we found out is there wasn't a frame shift that caused this. This was an ancient bacteria. People had actually published in literature in obscure language that they actually refuted that hypothesis. So this whole, for like 20, 30 years, even now, they cite that experiment and that claim as reality, and it's a myth, and it's still published in peer review. They say this is an example of a new protein. I'm just like, look, that was refuted in 1992.
Right.
John Sanford and I submitted to peer-reviewed publications, rejected 7 times. We finally gave up. I said, these guys aren't interested in truth. The editors
it's like what is it? Ontology or recapitulates phylogeny. That argument, the Bayer argument, Haeckel's argument, and that's been refuted, but it comes back that again and again, and then you press them on it, and this gets to the Mott and Bailey strategy. You press them on, they'll say, oh yeah, we didn't really mean that, and then 5 years later, you'll see a prominent person who admitted that it wasn't true, using it as an example of evolution. You're like, this was proved untrue, 125 years ago, and it just keeps popping up.
Yep.
And
Yep.
whatever.
And I'm just like, okay. So, I mean, what I was about to say, the editors would write us and say, look, we agree with you. You're correct. But we got a variety of things. It's like, well, it's out of scope of our journal, which is the polite way. He said, oh, but we already figured that out. This is nothing new.
Yeah, right. Yeah, right.
So we're rejecting on lack of novelty.
Yeah, right. Yeah.
And it's just like, okay, none of you said any of this, what we said was wrong. You just found a different excuse to get to not give us a hearing, even though, and you're just going to let this myth go on. I said, look, not even to make it personal, you could reject us, but why hasn't someone on your side called everyone else out on this? Because the whole industry is still citing this as a reality, and they're embarrassing themselves. And I just keep hope they keep citing that paper, because I'm just laughing. It's like, look, well, we heard from the editor that we were right, but this was old hat, so we're not going to publish. This is, discovered that it was a mistake.
Yeah, Wells's books on icons of evolution is really good at that, because I think he had a rejoinder to it where, the original response to icons of evolution was, he pointed out all these things like ontology recapitulates phylogeny. That is the development of an organism from, egg to adult is the same thing as, from the first organism to, man. And that she showed that, no, we've known for a long time that's not true. And then the response, he had like 20 other examples. And then the response was, well, of course, we don't really believe any of those. And then, 10 years later, Wells, came up with an update and said, no, you're still using those examples. You're still using those examples. And when you call them out on it, they say, well, we didn't really mean it. It's just like, whatever. The devil lies.
Yes. So, did you want me to help you at all, any with the anti-weasel project, or are you just wanting to share?
If you know anybody who maybe you could give me some feedback on it or what I could do with it. I'd just like to show it to somebody who might appreciate it. And, because if I just show it to random people I know, it's like what you're doing. It's specialized, right? So it's not an argument many people care about, or, care to influence, but maybe somebody who might appreciate it. And I think it's a good argument, but it's a niche argument. And this is like what you're doing. I mean, I'm sure like if you took a random person on the street and you said, well, okay, the arguments that you're pressing, how many people, what percentage of people are going to appreciate your arguments? Like probably 1 in 1,000, 1 in 10,000, because it's so specialized, right? So finding those people, since I'm not in the field, I'm just a guy who does this for fun on the side. If I could find, because if I just sent random letter like this to somebody in the field, it just goes to spam drawer, because it's like, well, I'm not going to read 3 pages by some guy I don't know. So if you can find someone who might appreciate it.
Well, I'll keep it in mind, and which sadly doesn't count for much because I'm really swamped with other research projects. But this is maybe I'll try to post it in a link to the video description, Do you mind me? Is it okay
Oh,
if I
well, let me
archive?
clean it up again. I'll run through my Grammarly again, just in case I have some typos.
Okay. And that would be fine. And then I'll update it to the video. I probably will get maybe 30, 40 views, but there's might be someone interested.
Okay.
Um, I am I wanted to tell you though, I'm very interested in some sort of anti-weasel project because this lack, Dawkins has done something. If it were willful, it was really dastardly because he's representing weasel as the way natural selection works. And that I said that uses a lot of foresight. And we know experimentally and also theoretically that's not the way this reproduction,
Right,
this
right. Yeah,
elimination of the least
yeah.
efficient. I said we have so many experiments that said this is not even
Right.
what happens in nature.
It's,
I
it's like
mean,
that evolution of like 1% increase in efficiency creates an eye. Remember that argument? Berlinski tore that one apart. It's like this really dumb argument where like you take a target and you move there like 1% every mutation, and you get there in whatever 300,000 years, and it's like, okay, but that's totally cheating. You're you know, and then they called them out on it, and then of course they just said, well, we didn't really mean it, but it still pops up.
yeah, and see, we have now experimental evidence. Like I said, all you needed is 5 nucleotides just to
Right,
recover a gene.
right,
One
right,
that
right.
I could probably
Yeah,
just broken one. I probably could have broken one nucleotide and it would never recover it. And I said, see, it never
yeah,
works like the way you say.
right,
Because I mean, now we have live experimental evidence, and
right.
I could just say I told you so. But I did want to show you one thing about the commonality just before I say goodbye. Um, People say, why did God make us so similar? I'm finding out, I mean, to other creatures, and why do we have patterns of similarity and diversity? It turns out all of these creatures like have a puzzle to understand human biology. Like the way we actually figured out how our genome worked, it began when we were studying bacteria
Right.
because humans are so complicated. It was easier to
Yeah,
work with bacteria to figure out how the genome worked. Um, when we wanted to study the nervous system, we would study roundworms, nematodes, and
right,
even squid. When we want to test out our pharmaceuticals, we tested out on mice and chimpanzees. And I said, this is so much better than if we had to dissect pigs. I mean, yeah, there's a picture of a fetal pig here. Let me see.
right.
See,
Yeah,
I said, don't you see? God has provided all these things so we don't have to suffer to figure out how, it says in the Bible, by his stripes we were healed. And like you have all these innocent lambs and other creatures that die
right.
in our place so we could find medical cures. I said, you see the metaphors of the Bible. And so I just wanted to say, so they'll say, oh, we're all similar. I said, well, I can make an argument that one, it didn't evolve. The similar, they'll say, why would the designer make it that way? I said, you ought to be grateful that you don't have to sacrifice your kids or any human to be able to do medical experiments. That's
Right.
what we have mice for. And that's why we ought to be grateful every day that there is a pattern of similarity. For the longest time, that was a stumbling block for me that I was thinking, God, well, why did you make chimps so similar to us?
Right.
And then now I'm realizing, thank God he did, because we have model organisms and we
Right.
do have even bacteria have some similarity to us, but that helps us understand. That's why we're able to find cures and also understand we're fearfully and wonderfully made.
Yeah, yeah, we are. It's just, yeah, it's good to, that's a great Christian virtue, gratitude. It's the beginning of so many virtues, but we really should be grateful. And it's a miracle.
Amen. Well, it was great talking to you, and
Yeah.
I thank you for being willing to share and be on video. You'll be, and I told my pastor today. I said someone wrote me a letter. He used to be an atheist, and now he's a Christian because he looked at the creation evolution controversy. And I said,
Yeah.
I'm actually meeting a lot of people like that. I said, see, they're
I
people
think
that
didn't Dean Kenyon do that too?
he may have been a Christian. There are a lot of people that were Christians and, in that era, but they believed in evolution. But he did believe he believed in biochemical, just self-creation. He called it biochemical predestination, not the theological predestination, but
Right.
he abandoned that. He said it made more sense to be intelligent design.
Yeah.
But the big conversion, one of the big conversions was Richard Lumsden. He definitely was an atheist.
Right. Yeah. I haven't met anyone who's become a Christian through intelligent design like myself, but I have to think there's some. But
Yeah, I've met them. There have been a few, and there are more and more as the longer I get to just talk to people, like Fuzz Rana of Reasons to Believe. He's an old Earth creationist, but he said he came from an agnostic family, and then he was studying the biochemistry of the cell membranes, and he said, "I think there's a God."
Right. Yeah, it's great. Yeah, it's good though, because like we have it's good for us to get together because most of the people I need who come to it do differently, and you can come various paths, but my process of sanctification it's been different than most people because I came from my head, not my heart, and it's a different set of things I have to work on and learn. Whereas for the people who came to it the other way, they have different issues, and so it's nice for guys like us who started with ID.
Yep. Well, I have to take off. Thank you for your time.
It's great talking to you. Yeah, it's always fun. I love talking about this stuff. I don't have like my Christian buddies in my Bible study. They don't, they're kind of interested, but they obviously don't want to go into the weeds. So it's really fun. Thanks.
All righty. Well, God bless you. You take care, and
You too.
Lord willing, we'll be in touch. Take care.
Okay. Bye.
Bye-bye.